Welcome to MicroMetabolism Database. I'm Tim Hackman, and in this video, I'll show you one of the many tools available through this resource. We'll focus on the Predict from Taxa tool, which will allow you to predict metabolic traits of microbes from the names of taxa alone. This tool can be useful if you've identified thousands of different microbes in your samples through DNA sequencing. You know the names of the microbes, but you have no idea what these microbes are doing. To use this tool, we click on the Predict from Taxa button here on the home page. We're brought to the tab for this tool. We see on the left where we'll submit names of taxa to predict metabolic traits. We'll do this by uploading a CSV file that has names. To see how to format the names, you can download an example CSV. Let's do that. You can see that there are columns for phylum, class, order, family, genus, and species. We can see that there's one row per organism. In this simple example, we have only 4 organisms. In each row, you enter as much information about each organism as you can. The first row shows an organism where all the names are known, down to genus and species. This is E. coli. However, we don't need to know that level of detail. Let's say you know only the name of the genus. That's pretty common in DNA sequencing of environmental samples. And that's the case for the organism in the second row, Streptococcus. In the third row, we can see that we've provided the family name. Again, that's okay. We provide as much detail as we can in each row, and we repeat the process for all organisms. After you're done entering names, you can save the CSV, then return to the MicroMetabolism database. After you've returned to the database, you can upload your saved CSV file using the file upload control at the left. After we've uploaded the file, the tool will automatically predict metabolic traits. Let's see the results we found in our example. Up top, we can see that we've predicted 3 metabolic phenotypes for our 4 query taxa. If we like, we can download our detailed results in CSV format. We'll return to that in a moment. Immediately below, we have pie charts that summarize our results. We see the types of metabolism in our first pie chart. We see that all organisms in our sample have either a fermentative or methanogenic metabolism. We can see even more metabolic traits to the pie charts at the right. Below that, we can see our results in detail. We can see the names of our query taxa, along with the type of metabolism and other metabolic traits. If you want to download these results, then return up to the Download Results button. This contains the same information as below, but in CSV format. There are a few options available for our tool at the left. To understand what these options mean, we need to first discuss how this tool works. It's similar to the tool FAPRTax. It first matches the name of a query organism in your CSV file to names of taxa in the database. If all matching organisms in the database have that trait, then the tool predicts that trait for your query organism. For example, if your query organism is from the genus Selenomonas, and all members of Selenomonas in the database have fermentative metabolism, then the tool will predict your query organism has that metabolism type too. Now let's say that we want to make this matching less strict and get more predictions. We can do that by setting the prediction threshold lower. Let's see what happens when we change the threshold from 1 to 0. We can see that now we've predicted more metabolic phenotypes, 5, because the threshold for matching in the database is lower. Now, if any organism matches our query, the database will predict a metabolic trait. By default, the names you provide should follow the taxonomy of Berge's manual. This is because the organisms in our database follow this taxonomy. If you want to provide names that follow NCBI taxonomy instead, you can adjust that using the radio button below. About 80% of the organisms in our database have NCBI taxonomy too, so you can match those. Many organisms in our database have had their metabolic traits predicted using neural networks. The accuracy of these networks is high, but not 100%. If you want to hide these predictions, You can do that with a radio button. To sum up, we've shown that from using just the names of microbes, we can predict their metabolic traits. This is especially helpful when you've done DNA sequencing, have names of microbes, but don't know anything else about them. By revealing metabolic traits, this tool can help you unravel what microbes are doing in the environment that you study.